everyone. Welcome to Thursday's lunch. As these wonderful young people, it's great to have young people among us, come up and take their seat. I just want to thank you for being here. And with that, I'm going to turn over the mic to Ramya.
Thank you all for coming today. I'm really excited to see you all. I'm going to keep my part brief because, as you can see, we have a lot of speakers today. As you all know, my name is Ramya. I'm the Director of Environmental Science for the Key Biscayne Community Foundation. We do these lectures once a month, specifically the Citizen Science lectures, and they're almost always the last Thursday of the month. That only changes if there's something going on, like next Thursday is Halloween, as you all know. And then November and December, they'll be a week early also because of the holidays. But otherwise, the rest of the year, last Thursday of the month, we always have some really interesting science lecture for you guys. So I'm just going to move on. This is Bianca. She's going to be moderating our panel today. I would just ask, each one of them has their own short presentation. I just ask that you hold your questions until the very end so that we can get through everybody's presentation, and then you'll have time to discuss everything you want with the panelists. I'll let Bianca introduce everybody.
Yeah, of course. Thank you so much for being here, everyone. I'm really excited to introduce all of our panelists today.
Hi, my name is Nicole Besemer, and I'll be representing the AOML Coral Program today.
Hi, my name is Megan Kennedy. I'm representing Rescue Reef through the University of Miami.
Hi, my name is Lisa Belskis. I am over at NOAA Fisheries, the Southeast Fisheries Science Center, and the Sea Turtle Program there.
Hi, I'm Jenny Ritz. I'm also at the Southeast Fisheries Science Center, and I'm here representing the Marine Mammal Branch today.
Thank you so much for being here today. So I guess we'll go ahead and get started with the first presentation.
Thank you so much for having me today. A little bit about me really briefly. I have lived originally from the Northeast, but I lived in Miami for over 11 years now. And for most of that, I've been a part of the Virginia Key Biscayne community throughout school, through work, and I lived right down the road on Galen for 2 years, and I loved it. So I'm really excited to be here talking to you today. And so I am going to try to give you a very whirlwind tour through our diverse research portfolio, trying to represent our entire group. So please bear with me as we rapid fire through this. And so just sort of as to get us started, our mission is to monitor, manage, and understand coral reefs in an era of global change. And so to try to tackle this, our lab has somehow gotten to 8 different research tracks. And some of the foundational work that really started our lab over 12 years ago is our long-term monitoring. And so one of our goals was essentially characterizing the acidification of coral reefs and our ecosystems. And so one of the places we do this is actually one of our favorite reefs and really important reefs. It's called Chica Rocks, down off the coast of Islamorada. And this is a buoy that we have installed and managed for the past 12 years that provides near real-time data on pH as well as concentration of CO2 in the water. And you can go to a website anytime to access this, so it provides really great sort of baseline environmental data. We wanted to expand sort of our reach and where we were doing all of this really important long-term monitoring. And so the project that I coordinate is the National Coral Reef Monitoring Program, or NCRIM. And so we do climate monitoring specifically. So we use a lot of different instruments that monitor temperature, bioerosion, calcification, and we visit all of these sites throughout the U.S. Virgin Islands, Puerto Rico, dried tortugas, flower gardens, and so we're able to provide all this really baseline data of trends and also just status of coral reefs and how they're doing as time goes on. Another important aspect that we kind of wanted to look at is the concept of extreme reefs, and a lot of these are in our backyard right now. One of them is the Port of Miami. There is quite a variety of corals that actually live right on the seawalls and on the riprap right here in our backyard. And so we wanted to be able to take this information. These reefs experience really diverse temperatures, very extreme changes in temperature, salinity, and they're surviving, they're thriving. Sometimes they're even out of the water. And so how is that, right? And kind of gives us a little bit glimpse into these future conditions and what can we learn from them. We also try to look at reefs that are sort of in the more traditional reef habitats, but doing really well. And so this is more pictures of that reef I mentioned in Islamorada, Chica Rocks, which is beautiful. It has some of the highest coral cover throughout the different Mission Iconic reefs in the Florida Keys. And so we're also looking at these different, basically future conditions that we could see. Climate scientists have produced these different models of temperatures we're going to experience. And researchers from our labs are basically fitting those models with the data that we have and seeing how is this reef that's doing really well now going to survive, especially if we can do things like either mitigate some of the climate change impacts, or if we can do things like restore our reefs, like our partners that you'll hear about, Megan, to be able to like put put corals back out on the reef and how they're going to do in the future. We also try to take the corals and try to understand their molecular mechanism. So what's kind of going on in that coral, whether it's different types of genotypes or what gene expression is happening or microbial communities that are actually helping them to survive better in some conditions than others. Another aspect that we focus on is coral disease. Some of these diseases are really well known and have been around for a while. A more recent coral disease, stony coral tissue loss disease, really started in Miami back in 2014, and scientists still haven't figured out essentially what is causing this disease and the best way to kind of mitigate it and stop it. So we're ever trying to work on that. One of the most important things we do is kind of take all this information and we want to figure out how that can actually help and basically our restoration practitioners. So these people like Megan who you're going to hear about that are out all day, every day, out planting corals, putting them back on the reef. We want to provide that information of which coral might do better in this type of environment or stressor or might survive better against a disease. And so to really make sure that the work we are doing is the most efficient. And last, which is very far from my realm, but I think is the most interesting, is we've gotten really into engineering. And so none of us are actually engineers by trade, but it's become some kind of a learned practice, and we're trying to figure out ways how to use robots to feed our corals and clean our tanks and become more efficient. So it's fun when we like buy these robot arms, we name them after our staff members, and they're like dancing around, doing crazy things, high-fiving them and stuff. But they are really important, and we're also able to not only do that, but then create our own computer systems that help manage our coral tanks, and we're able to run these experiments. So I think sort of that really open source innovation and design is really important as well. And while this sounds like a lot that I just rushed through, all of these are really connected and really important to help preserving our coral reefs. So this is our amazing large team. Dr. Amy Knox is the principal investigator up in the right-hand corner. And thank you for listening to me today.
Hi everyone, I'm Megan Kennedy. A little background about me, I'm from Florida. I did my undergrad at Florida Atlantic University, my master's at Nova Southeastern University, and then I moved down to the Keys to do coral restoration, and now I'm at University of Miami as the program manager for Rescue Reef, which is what I'm going to talk about today. So who here is familiar with corals and coral restoration? Okay, amazing. Okay, a couple of people. So this is some new information for you guys. It is a shortened presentation, but you're going to get a ton of information. So this is restoring the future of coral reefs. So corals are super important. They're actually animals. And they're important to us here in Miami because they provide coastal protection. They break up wave action when we have hurricanes and storms, and that helps protect our coastlines. They're also habitats for reef organisms. Does anyone here like seafood? Eat seafood, lobsters, fish? There's a good chance that that food has actually spent some portion of its life cycle on the coral reef. So this is really important to us. Also medicine, it provides jobs, fisheries, and tourism. Tourism is a huge driver in Florida for our economy. And this actually relies on stony corals, so corals that secrete a calcium carbonate skeleton. So they're hard corals, and that's what's important to us. And those are the corals that we work with during restoration. The coral reef crisis is something that has been ongoing essentially since the 1980s when we started losing herbivores. Herbivores are important because they eat the algae that outcompetes coral. So some examples of that are sea urchins. There are also some fish that eat algae. As Nicole was saying, disease is pretty prevalent. Hurricanes can topple corals over. Bleaching events is actually when the water gets really warm and the corals get very stressed and they release their symbiotic algae that provides energy for them. And then they just rely on grabbing food out of the water column, and it's just not enough to sustain them. So if the waters don't cool back down, the coral can actually die. And this is happening more and more often and with more severity every single year. Predation are fish that eat corals, as well as what's important to me is shifting baseline syndrome. So somebody who just got their dive certification and they go to the reef and they're like, this is amazing, there's corals everywhere. Well, 50 years ago, there was 60% more coral. And today, there's just less than 5% coral cover left. So that's why we have Rescue a Reef. This is a citizen science program that is created through Dr. Diego Lehrman's Coral Restoration Lab at the University of Miami. And we actually do citizen science, where we bring citizen scientists out on the boat with us, and they get to work with the corals themselves. They actually help us do research, like performing surveys, and restoration by outplanting the corals. We work from Sunny Isles down to Biscayne National Park, and primarily we work with staghorn corals, which are seen there. We have 3 offshore nurseries, and nurseries are where we grow corals on PVC trees. And these PVC trees are taller than I am, and we use fishing line to hang the corals on there. They grow, we cut them, we bring them to a new tree, they grow, we cut them, and this is how we farm them, essentially. And when we want to outplant them to the reef, we cut them and we just swim them over to the reef instead of putting them on another tree. So citizen scientists have outplanted more than 40,000 corals for Rescue Reef since 2009, which is a pretty big number. And they're all going right offshore of where you guys live. So our citizen scientist expeditions, the goal is education. So we do a briefing, essentially like I'm doing with you all, about corals and why they're important and what we're going to be doing today. And then engagement, which is where people can actually go do it for themselves with their own hands. And then transformation. We want people to go home and tell their friends and family about what they did today and spread everything that they learned about why corals are important and what's going wrong and how we can fix it. So this is what dive 1 looks like. These are the PVC trees I was talking about with the corals hanging off of them. And the first thing we do is actually clean the trees. It's important to clean the trees because, again, algae competes with coral. So we want to make sure the trees are always clean. Our corals have the best chance of growing as fast as possible. Well, on dive 1, we also collect the corals, like I was saying. So we use snips, actual metal snips, and we cut off just fragments of the coral, and this actually makes the coral grow faster. So the coral that's still on the tree is growing faster, and the coral that we just grabbed to bring to the reef is growing faster too. And this is a natural process that normally happens by hurricanes and, you know, animals that bump off coral or something like that, but we're just taking advantage of that in a more controlled setting. Then we actually put them in laundry baskets, and then that's how we transport them. So in between dive 1 and dive 2, this is topside. So we use cement, we mix it on the boat, which the dive shops love that we do that on their boat. And then if you're a snorkeler, because we do bring out snorkelers, we have these coral cookies where you can actually outplant pieces of the coral on the boat, and a diver will bring your coral cookie down for you. And this is dive 2. So this is where we bring down the cement piping bag, and this is how we actually secure the corals to the reef. And the corals that we put there, we put in a lot of care into making sure they're in a good spot, because once we cement them down, this is essentially where they will live the rest of their life. So here are citizen scientist impacts. We have over 10 restoration sites, over 100 expeditions, over 1,000 citizen scientists, and again, over 10,000 corals, but it's just nice to go 10, 100, 1,000, 10,000, so that's why it's structured like that. This is what it looks like right after we outplant. So you can see the little coral fragments there. And then this is about a year later. So they grow very fast. We do take advantage of the staghorn coral because it is a keystone species here in Florida. If you were an urchin or a fish, wouldn't you like to live in those branches, you know? And we also have what we call a community coral garden project. And this is done at Paradise Reef, which is just a couple miles offshore from here. And essentially what we're doing is repeatedly outplanting to the same place so that we can try and understand what the impacts are of rapid restoration. But it's not just outplanting, it's predator removal. There are fish and worms that eat corals, and we want to get rid of them. Well, not the fish, we like the fish, but we do remove the worms because they spread disease and they injure the corals, and they shift the corals from growing to healing because they're being hurt. We also do coral monitoring, so the bleaching we were talking about, the disease. We want to write that down, we want to understand it, we want to give it to the smart people at NOAA so they can tell us what's going on. And we also do offshore cleanups where we bring the garbage out of the water back onto land, dispose of it properly. But it is important for people to understand that it's not just environmental factors that are affecting our coral, it's also us directly affecting them as well. These are some of our sponsors.
Okay, I have to follow that, and I definitely want to volunteer to pick up some of that trash. So I'll definitely contact you about that. Again, my name is Lisa Belskis, and I have my little notes here in case I freeze, because I know myself. And anyway, so I have been working for the Southeast Fisheries Science Center for, a long time. I guess 26 years, started in 1998, and was hired as a full-time federal employee in 2002. So my role has changed over the years quite a bit. But right now, currently my role is split between being a research fishery biologist for the Marine Mammal Turtle Division, and the other half of my time is spent as a communications specialist for our center's director office. So I am just one member of a dedicated team in our sea turtle branch. And so this is us. Here we are, all smiling, doing our work. I didn't get permission to use their images here, but it has been used in plenty of other presentations, so I think it's okay. But we are a relatively small group. Believe it or not, 8 people is a small group in this type of research. But we cover a lot of different topics, and these are just some of the things, and I know I missed plenty, like distribution and mentoring and all of the other things. So what we do as a sea turtle program, we conduct a science that leads to improved knowledge and meaningful conservation of sea turtles and their habitats. So that's our goal, that's what we do. And we all do it to support the Endangered Species Act. That's the legislation, the foundational legislation that allows us to do this work to conserve sea turtles. So NOAA is one of 2 federal agencies that is responsible for the protection, conservation, and recovery of sea turtle species and other species that are listed under the Act as well. The other federal agency is U.S. Fish and Wildlife Service, and they're responsible for basically sea turtles on the land, and NOAA is responsible for sea turtles in the water. So here are 6 of the ESA-listed sea turtle species. We do research on 5 of these. The 6th, the olive ridley sea turtle, is very rare in our waters. We have encountered them in the stranding record and in one of our monitored or observed fisheries. But they're very rare, so we don't really concentrate our efforts there. So we concentrate mostly on leatherback, loggerhead, green, hawksbill, and Kemp's ridley turtles. And in Biscayne Bay, right in our backyard, our most common sea turtle species that we encounter and we do research on are green and loggerhead turtles. And by far, they are the most abundant in this area of all the sea turtle species. We do also encounter hawksbills and the rare fusion of Kemp's ridley turtle, but for the most part, like I said, we do most of our research on green and loggerhead turtles. So as our program, we do believe that the health of sea turtle populations depends on advancing our scientific knowledge and gaining more information from us on the science side, but it also takes a lot of ocean stewardship, and we all can participate in that. The information we collect through our work allows us to provide science-based technical advice to fisheries managers and other ecosystem managers. And we support this by providing information for sea turtle assessments and characterizing the direct impacts of threats. So I was asked to provide what my role is at Fisheries, and it has evolved quite a bit. But when I first started with sea turtles in general, it was back in 1984, and that's the first place I was permitted to work with sea turtles. And of course, any protected species, you are required to have permits. And it was so exciting when I got on the first permit. And I haven't been able to get off since, but it's been wonderful. So I first started on Nesting Beach, just north of here in Broward County. And then when I came to NOAA, I began working as the coordinator for the Cooperative Marine Turtle Tagging Program. And that was a program that would distribute and enter data that would distribute tags, so the metal flipper tags that get attached to the leading edge or the trailing edge flippers. And I would distribute those to folks in the Atlantic Ocean Basin, ensuring that there was no duplicate of a tag series, making sure that data got entered, and then reporting back to any any project manager when they happen to get a recapture and sharing that information with them. And then some of the most extraordinary experiences I've ever had, and luckily it still happens occasionally, but I've gotten to do some NOAA ships, or the really large white NOAA ships, and small boat vessel surveys, and I've also done aerial surveys, which the perspective from either of those places is phenomenal, and some of the most amazing things that you would ever see, in every trip that I've ever been on has something memorable about it. That has been super exciting. So I pretty much started with doing something like that, and occasionally I still get out in the field and able to do that. For the most part of my career, I've worked with the Sea Turtle Stranding and Salvage Network, and many of you may have even participated in this on Key Biscayne or other areas of the coast. So the Stranding Network is basically from Texas to Maine, and volunteers, network participants throughout all of those states in the coastal waters have reported sea turtle strandings. And you can have a stranding that may be alive, it may be dead, it may be sick, it could be injured. There's lots of ways sea turtles strand, and it happens quite a bit. So I wouldn't doubt it if you guys, some of you aren't already participants in that network and are very familiar with it. So another role that I've played is training seagoing observers. And so the observers may work on a number of different fisheries, but it's out there to observe fisheries. And so I would train the observers to make sure that they can identify the species correctly, how and where to measure turtles, how to tag them, and of course document them in the way that we needed to know. And then also some sample collection is done out in the field as well. And then lastly, I have transitioned. Like I said, half of my time is now spent with the Southeast Fisheries Science Center's Directorate Office, and I spend that highlighting our center science for all of our center and all of the things that we do. And I basically work on the webpage, but it can be social media, blogs, website, and things like that. And I wanted to thank you for letting me share a little bit of my journey about sea turtles and NOAA. Thank you.
All right, thank you all again for having me today. So again, my name is Jenny, and I have been with the Southeast Fisheries Science Center, like Lisa, for more than 25 years. We were like 10 when we started, really. I also came down originally to Miami for undergraduate work there, and then I got my graduate degree at the Rosenstiel School as well while I was working at NOAA. So I've been within the marine mammal group for a while. I'm now the marine mammal branch chief. And these are the people, though, that do all the hard work. This is our team. We have about 25 people in the marine mammal branch that run from student interns all the way up to PhD level researchers. We have veterinarian, we have a veterinarian, we've got acousticians, geneticists, marine biologists, modelers, statisticians, a big diverse group that does all of this work. In the marine mammal branch, also our goal is to promote the stewardship of marine mammal populations through science-based conservation. So as Lisa said, we collect the science and the data, we provide it to the managers. A lot of what we do is to delineate the different marine mammal populations, look at the population boundaries, where there are overlapping populations, understand how many of a particular species are there, is their population increasing or declining or stable, what are the impacts to those populations, particularly from humans, but also how is their health, cause of mortality, risk assessments for different impacts, investigate mitigation measures to try to protect or recover marine mammal populations, particularly those impacted by us. That top right picture you might recall is the Deepwater Horizon oil platform explosion from 2010, and we are still to this day researching the after effects of that spill and how we can help marine mammal populations recover from that spill. And now we're also investigating and planning for current and future potential impacts from climate change. This is our toolbox. We do different surveys depending on the habitat. So if we're looking at the oceanic species, we go out on these large vessel surveys for 2 to 3 weeks at a time. If we're looking at coastal and bay species, we do small boat work, and we tend to fly aerial surveys over the coastal and continental shelf water. Other tools we use are biopsy work, tagging, photo identification. We have a passive acoustic monitoring program that use instruments both off the large ship to listen to whales and dolphins, but we also have moored instruments that we put out in the water to record that acoustic data. We have a genetics laboratory that looks at population genetics, eDNA, other omics work, species identification. We have the marine mammal stranding and health investigators, much like Lisa talked about on the sea turtle side. It's the same thing for the marine mammal side, responding to those strandings and trying to determine what caused their illness or their death. And as I mentioned, many analytical tools and modeling tools. This is our jurisdiction is the NOAA Southeast region, which starts at North Carolina, comes all the way down the Atlantic coast around Florida over to the Texas-Mexico border, and includes the U.S. Caribbean, Puerto Rico, and the U.S. Virgin Islands. And it encompasses all of the U.S. federal waters out to the U.S. EEZ. You'll notice I'm only talking about dolphins and whales here. We do have some seals in the North Carolina area from time to time. I will not talk about manatees because they actually are covered under the U.S. Fish and Wildlife Service jurisdiction as well and not under NOAA's jurisdiction, but we work with a lot of the same partners. You might be surprised to find out how many species of dolphins and whales we have in our U.S. waters. Most of you are familiar with bottlenose dolphins. That's what lives here, close to shore. That's what you'll see in the bay. But in the offshore waters, we have multiple species of endangered large whales. Primarily, we work with Rice's whales, which I'll talk about in a minute, North Atlantic right whales, and sperm whales. There's also a lot of other species of smaller whales and oceanic dolphins that you might be familiar with. Pilot whales, beaked whales, killer whales, spinner dolphins, spotted dolphins, and so on, until we get to about 26 different species. This is the Rice's whale. I would like to introduce you all to the Rice's whale. Many of you may not have heard of it, and there is a reason for that. It is the only resident baleen whale that lives in the Gulf of Mexico. It is highly endangered. We believe there's less than 100 of them left. However, for many years we thought it was a Broodies whale. Broodies whale is a different species that lives worldwide. And it was only in the last 10 to 15 years that we started to figure out it was actually a completely different species. We first discovered that by looking at genetics, but in order to name a new species, you need two different forms of evidence. So when one of them stranded, unfortunately dead in 2019, we were able to collect the skeleton and the skull and work with the Smithsonian and other partners to show that there were actually morphological differences in the skull. That gave us 2 lines of evidence to describe this as a brand new and unique species. To date, we think is primarily in the Gulf of Mexico. Again, less than 100 of them left. So we have quite a big learning curve. We've made a lot of strides in the last 5 to 10 years learning a lot about this whale, but we have a long way to go and a lot to learn. So to that effect, I put some handouts on your tables. There's a handout on the Rice's whale if you want to learn more. Also, I'm going to tell you this is hot off the press. We don't even have our website out yet, but we just finished a very successful tagging survey in August where we were able to deploy 7 suction cup tags on 7 different whales, which will give us some really exciting data on feeding ecology, on dive behavior, and calling behavior. So this was working with partners, the Ocean Alliance, to test a new tagging technique. You'll see on the top left there is a drone, and that orange spot is a tag. So this is a new technique where they can fly the drone over the surfacing whale. The drone lets go of the tag and it plops on the whale's back. You might see that orange spot in the top picture there. That's the suction cup tag, and it stays on anywhere from a few hours up to 18 or 20 hours. Then we go pick up the tag and get the data, which is super exciting. The other species I want to talk about is just bottlenose dolphins, because that's the one everyone's most familiar with. They are close to shore and most impacted by humans. The picture on the bottom is freshwater skin lesions from animals that were exposed to salinity drops in a bay in the northern Gulf. So one of the climate change areas of research is the effects of fresh water on dolphins from increased rainfall, increased runoff, and increased storms. Here at Biscayne Bay, we've had a photo ID study going on since the mid-90s. We do photo ID by collecting photographs of dolphins' dorsal fins, and you'll see they can all be a little bit different, which lets us follow animals over 20 plus years. Our recent estimate is there's about 240 bottlenose dolphins in the population of Biscayne Bay. They have a social structure within the bays, so the ones that tend to hang out in the north hang out with the other ones in the north, and then we have another group in the south. they kind of intermix in the middle. And we found that the animals that spend most of their time in northern Biscayne Bay have very high levels of PCBs and organic contaminants, which we also find in sediments in Biscayne Bay and prey fish in Biscayne Bay. Recently, we started a partnership with FIU and some of their students. They're investigating the feeding ecology of Biscayne Bay dolphins and responses to habitat degradation, in particular loss of seagrass beds in Biscayne Bay. These are some of the stressors facing bottlenose dolphins and really all of the marine mammals now. In coastal waters, it's a lot of boat harassment or feeding, fishing line entanglement, and in the offshore waters, ocean noise, and others. And I'll leave you with this slide of what you can do to help marine mammals, and I've also put some cards on your table if you like to go boating, if you are a fisher. There's some dolphin-friendly fishing tips and boating tips on the table as well. And that was about as fast as I could go.
Thank you so much, everyone. I open up to the room if anyone has anything that they would like to ask any of our panelists. Oh, here in the front.
How was the turtle egg season this year?
The question was, how is the turtle egg season this year?
I can answer that. I do see, as I think I've told you guys, I do see turtle nesting at Bill Bags, and they also, it was very average year, yet last year it was insane. Just the numbers were really, really high, crazy. And this year the numbers really just followed kind of that median line that's like almost exactly average. And that's been the case throughout most of the state. I think it was the green turtle nests were a little higher than the average. But everything else was pretty much right along a normal average year.
What conditions change?
No idea. There's a number of things that might go into that. A lot of the turtles have specific years that they'll have a higher year and then like a lower year. Like it depends on the species. They'll have a higher year, a lower year, a higher year, a lower year. Some of them have 2 lower years and then a higher year, something like that. There's also some speculation that it might be due to the pandemic, and a lot of people weren't out of the water. There was less going on, so they were able to feed better maybe during that time. And so that's just a theory, but you know, we don't know really. Those are just some of the reasons that I've heard about.
Right here?
So what's the impact of the shipping industry, both cruises and container ships in the ecology, marine ecology, whether it's coral reefs, marine life, I don't know. I mean, especially now that they have larger and larger ships.
Yeah, of course. So the question was, what's the impact of the shipping industry? So cruise ships, cargo ships, all those bigger ships have on all the topics that we talked about today, corals, animals in general. So that was the question.
One of the things that's happened in the past year and is going to be a problem moving forward is especially when new cruise ships and bigger cruise ships, and so they need to make the port bigger, so the port needs to be dredged. It's really stressful on the corals. When they dredged the port, I think it was 2013 in Port of Miami, they had to relocate so many corals, so it displaced a lot. There was a lot of sedimentation issues that actually killed a lot of corals. And I think they're still kind of in the process and going through it, but they're about to do this up in Port Everglades. So it definitely causes quite a bit of stress to the coral community.
The big risks and our concerns, particularly for the large whales, are vessel strikes. Particularly in the North Atlantic right whale world, there's a lot of work going on right now on reducing the risk of vessel strikes, and we've been involved in some of that modeling, the risk assessment modeling. So there's some rules in place as the right whales migrate down to the southeast in the winter. In the various places around their path, there are some speed restricted areas. So there's a lot of work going on right now on trying to mitigate the risk of vessel strikes for right whales, and we are starting to apply some of that to Rice's whales as well. We have seen one Rice's whale in our catalog that does have evidence of scars from a vessel interaction, and we did have one that's stranded dead on the bow of the ship. So it does happen. It is a risk factor, but you know, our job is try to mitigate those risks, because obviously there's a lot of our economy also that relies on ships and vessels, and so we do our best to try to find ways to mitigate that. The other factor of that we often don't think about is ocean noise. The Gulf of Mexico is one of the noisiest ocean basins in the world, and so we also have some work in our acoustics team that's monitoring ocean noise as well, and that can lead to be part of a restoration project where they're gonna try to work with some shipping industry partners on voluntary measures to try to see if there's ways to reduce the ocean noise as well coming out from those ships.
You said that they're only local within the Gulf of Mexico. They don't travel, as far as you know, outside the Gulf?
Yes, so to date, we have found them in the Gulf of Mexico. We've more recently heard them on acoustic monitoring devices in the Mexican waters of the Gulf of Mexico, but we have not heard them on the Atlantic coast yet, although there have been some confirmed Rice's whales that stranded dead on the Atlantic coast. So, you know, they might have been out of their habitat, we're not sure, but for the most part, we have been seeing them and hearing them in the Gulf of Mexico so far. There's a lot of. How do we do the population estimates? That's a great question. A lot of what we do is from those visual ship surveys that I mentioned, or aerial surveys where we go out and travel. They're called line transect surveys. You travel along certain lines. You see a certain number of whales on that line, and then you use statistical modeling to extrapolate the number of whales you saw out to what's potentially in the area. That's how we do the abundance estimation for a lot of the species in oceanic waters. The Rice's whales are particularly challenging because there's so few of them. It makes it harder to do that modeling. So we have a need to do some more surveys. We also have photo identification catalog we've started for Rice's whales that identified a certain number of individuals, and then if we were able to collect a biopsy sample, then we would have genetic sequencing as well to see genetically distinct individuals, so we can add all those up to a minimum number of whales. But that's why we kind of say we believe there's less than 100 because the actual most recent abundance estimate is 51, but we know there's a degree of error on either side of that.
And it's not just scientists, it's linguists and the whole.
I'm not familiar with that project. She mentioned that there was an article that talked about the vocalization and the linguistics of whale communication. We do study the whales' sounds so that we can determine species and number of species. We know, for example, in bottlenose dolphins, if individual dolphins have a signature whistle that they use like a name, but I don't know about communicating with them. That's not an area that I'm involved in. Our team on the acoustic side is using it to identify species and record what species are where and how many, and more of those types of questions. You did ask me about the size of the brain, and they do have a brain to body size is large, in proportion to the size of their body. Yes, they do have large brains like us. I like to say that I think in some ways they're smarter than we are in their own environment, but they're very smart, yes.
Do you microchip them or do you use the drones in order to track them?
The question was for the whales in the tagging, do you use the drones where it pops on the suction, or you put an actual tag on them?
So for different species, we use different types. We've not done any microchipping in the blubber. You would need a reader for that. For the large whales, especially Rice's whales, are so highly endangered. So far, we've used the suction cup tags. There are implantable tags that go into the blubber layer that can stay in longer. We've not used those yet on Rice's whales. They have been used on other large whales, and those tags will then stay on a bit longer, a few months before they come off. On bottlenose dolphins, we have radio tags that you can put on with an antenna and follow them using radio transmitters. We also have satellite tags that can be attached to the dorsal fin. We also use photo ID techniques as well.
There's a whale in one of the canals here in Biscayne.
So the question was that someone said they saw a whale in one of the canals on Key Biscayne, and whether or not that can happen.
So it can happen if they are stranded and out of habitat. So other than bottlenose dolphins, no other species of whale or dolphin should be here in the canals. So if you see something that's not a bottlenose dolphin that close to shore, it is likely ill or dying, and we would ask you to notify the stranding network. We have the 1-877 whale helpline, or you can call your local law enforcement. They'll notify the stranding network to come and check out the animal and see what's going on. But we do have, I responded here on 4th of July one year to a pygmy sperm whale that stranded on the beach. So we do occasionally have whales stranded here. There were also some offshore dolphins that came into one of the canals around here once in past years.
To be threatened by sharks, the dolphins go around and protect them. It's unbelievable how they know when they're sensitive to whatever interaction with humans dolphins have. So
not part of the work that I have done, because I've been focused more on health and cause of death and those kind of things, but I know there are some groups that do. Dolphins Research Center and Dolphins Plus in the Keys, for example, do some of that research, and they have some programs with people that might have different abilities that swim with the dolphins, and they do some of that research. So there are groups that do that research, but we don't do that research at our office.
So this is for both of the coral people. I actually just recently gave a lecture at one of these monthly lectures about keystone species. And I did mention elkhorn and staghorn coral. Since I was going over all different types of keystone species, I only spent a couple minutes on each. Can you guys kind of talk a little bit more about why the elkhorn and staghorn corals are so important and what makes them a keystone species?
Okay, so elkhorn and staghorn are named based on the way that they look. So staghorn looks like a stag's horn, elkhorn looks like an elkhorn. And it's like I was saying in my presentation, they create, naturally and historically, they create these thickets. So it'll be a ton of corals tightly packed together, and it creates a really important environment for urchins and fish and other crabs, things like that. And it helps create the 3D structure that reefs need to become a coral reef, essentially. So like the soft corals, those aren't really helping anybody. And then, yeah, there are other hard corals, like bouldering corals, and those are going to help us break up the wave action and things like that. But yeah, those are, elkhorn and staghorn are two species that are super susceptible to the water temperature changing and to threats, anthropogenic threats and things like that, and they have a harder time bouncing back, and we think that that's why we've lost so many of them compared to some of the bouldering species.
Yeah, I was just gonna say the branching type, as we're mentioning, are the fastest growing. So they're actually the ones that are able necessarily to like respond quicker, or a piece falls off and then that piece can attach and like create a new coral really quickly. Whereas some of the more bouldering ones, which provide sort of different, as she mentioned, those ones are much slower growing over time, but those are the ones that contribute to the larger sort of like whole reef structure that's really keeping a lot of that shoreline defense. So it kind of is a couple different like areas, but the specific to the branching ones are going to be like the fastest responders and kind of help get that reef back up quickly if something were to happen to it.
I have one question for anyone who can answer on the panel, and that is, what is one misconception that you encounter a lot in your job that if you were to just explain right now or clarify it, you would do that right now?
I can go first. Sure. I think one really common misconception about coral is just based on the way that they look, and people think that they're plants. But they are actually animals. They eat, they have a gut, they can sexually reproduce, so they can send sperm and eggs into the water column to mix, and baby corals can actually swim, so they'll swim down to the bottom and they'll attach themselves and they'll grow into a polyp, and that polyp will turn into more and more polyps, and then you have a coral colony. So they are animals, and yeah, I think that's a pretty common misconception.
One I'll add is not really specifically about the animal, but people always think like, oh, you become a marine biologist, you spend all your time in the water diving, this and that, and it's probably like 15 to 20% of my job, and the rest is just paperwork behind a computer.
I would say I hear a lot about straws, right? Getting rid of, and we should get rid of straws, plastic straws. I agree, but it's not just straws. It's balloons, it's plastic bags, it's all kind of marine debris. And I'll say that I could be on a ship 200 miles from shore and a big batch of balloons floats by, and it's just the saddest thing. So I think there really just is a strong need for all of us to reduce that marine debris and plastics in general, not just the straws for the sea turtles.
Straws was a pretty big one here. Straws was a big one, yeah. It was a big one here. I guess in general, working for NOAA, people always ask me about the weather. So I'm typically asked about the weather, hurricanes, what's happening, but I have absolutely no idea. I leave that to anyone else.
Thank you so much for contributing your time here today, and thank you so much everyone for being here.